Okay, so now that the alarm is actually off, one of the things I might want to recommend, by the way, because, it's now, 12:35. If you want to write a thank you note to my dad, who's currently watching my children sleep, so I could come into Eugene to be able to do this, that'd be fantastic. And stuff. He'd probably really appreciate it. Anyway, so here we go. This is the second part of this. I thought I could pause and come back to it. I can't. So here's part 2. Alrighty, so here we go, ladies and gents. You might have to pause this for yourself because I'm going to go fast because I want to go home. Have a wonderful evening, of course. So here we go. One of the first things is when we're doing this process, this right here, and I left it up here. I'm just going to get rid of it, okay? Instead of having the light-dependent reactions up here, that's what these are. These are the light-dependent reactions, okay? So that's these right here. Follow that by this process down here, the light independent, and I did totally steal a little picture. This one I drew myself. Anyway, so the light dependent reactions, you're going to need to be able to do that is you're going to need some light energy coming in. That light energy is going to come in and get pigments all excited. So remember how we talked about that the photosystems are not only a thing but a process? So this is trying to capture both of those on here, okay? So photosystems 2 actually occurs first, followed by photosystems 1. If you ever want to know the little story behind that, I can tell ya. Anyway, at least the story that I've heard of why it's done that way. Anyway, so we got photosystems 2. Let's talk about the thing first. This thing right here has 2 parts. It has in the middle here what's called a reaction center. Okay? In that reaction center, so the center of all amazingness, right? You have chlorophyll A, P680. Remember, this has to do with what wavelength of light in the light spectrum it absorbs the best. Does it get the most excited with? Okay? Now, surrounding that, you have what's called the light harvesting complex. Okay? That light harvesting complex, because it's harvesting light too, that's the reason we call it that, okay, and it's complex because it has 2 different pigments to it, okay? In that, there is chlorophyll B and the carotenoids or carotenoids, okay? Those 2 are what we call accessory pigments, okay? Remember, we've got chlorophyll A is the primary photosynthetic pigment. Then you have chlorophyll B and the carotenoids, which are accessory pigments, like being an accessory to a crime, right? They're helpers in this process, okay? And so chlorophyll B and the carotenoids also get excited because of the lovely light energy. So these guys get excited and transfer that excitement to chlorophyll A in the form of electrons, okay? So those little electrons are going to go from these guys, and also when light energy hits here, it's also going to pop off a little electron of the chlorophyll A. So it's going to destabilize because it gets all excited and goes bing, and there it goes. It's a little electron too. Ready? Ready? Totally amazing. The primary electron acceptor molecule. Okay? So, which is an actual thing there again. These are embedded within the membrane. Okay? The problem is, now chlorophyll A, P680, flung off its electron. It's like, oh no, I don't have an electron. I need one. Where is it going to steal it from? The most ubiquitous thing that's in your body, water. Okay? That water molecule is going to be stripped of an electron to be able to stabilize these pigments. When that happens, it splits it. Because light is involved, we call that photolysis, also known as photolysis. So photolysis, breaking water with light, okay? When you do that, that's going to split off. When you strip those electrons, you're going to get some oxygen out of that process, which the plant can use for aerobic cellular respiration, and thank goodness a lot of it goes out through the spongy mesophyll, out the stoma, and we get to breathe it. okay? And then also out of that, of course, because you look at the water here, you're also going to get some hydrogen ions. Quite beneficial because those are going to get used right here, okay? So what we have next is we've got this chlorophyll A P680 that has gotten excited from light, as well as its little buddies here who have also gotten excited and shared the love. So everybody gets excited. Chlorophyll A throws off an electron that gets caught by this molecule right here, the primary electron acceptor molecule. And then that electron gets bumped from one to the next to the next. So what we're doing here is I'm showing you what we call that non-cyclic or linear electron flow. The electron from chlorophyll A zoomed up here, ended up on that primary electron acceptor molecule, but then it passed it on to another molecule, and then another molecule embedded in the membrane, and another molecule, and another molecule. That's why it's called the electron transport chain, because it's a chain of proteins in a row, okay? And so this is, of course, embedded within that membrane, and that membrane being that thylakoid membrane, okay? So this membrane is embedded with proteins that as soon as they get these little electrons, they get all excited too. And when they do that, they're going to transfer hydrogen ions, okay, from out here in the stroma, okay? Here's your thylakoid membrane, okay? And so this thylakoid membrane with the stroma on one side and the itty-bitty living space on this side. So this is, over here, is going to be your thylakoid space, is where these electron transport proteins are going to start pumping from the stroma into the thylakoid space, the inner membrane space, hydrogen ions. Okay? So, the other thing I want you to do is kind of go like this and like this. These are also membranes because this right here, this thing that looks kind of like a mushroom, right, that's just starting to bud, the basidiomycete, is ATP synthase, exactly the same one as in aerobic cellular respiration. So synthase, because it's an enzyme that synthesizes ATP, okay? So, electron transport chain during photosystems 2 starts getting all these hydrogen ions moving from the stroma into this itty-bitty living space. That creates, right, that electrochemical gradient or membrane potential. When this happens, that means that ATP synthase can allow these hydrogen ions to come in through it and then go out here. So here's my little hydrogen ions being able to go out through and back into over here, which is the stroma. Okay? When that happens, this chemiosmosis looks familiar, doesn't it? Okay? If I get rid of stroma and I go like this, itty-bitty living space with an electrochemical gradient and some ATP synthase, it's going to do chemiosmosis. So you're going to take some ADPs plus some phosphates, and you're going to then utilize these running through like the little turnstile, you know, at say the metro or subway, right, is going to get you then ATP. In this case, we're not going to do any numbers along with this right now. We're just going to do the big overall chunks, okay? So, Photosystems 1 is making and generating ATP through that process. But that's not all, because now we have a problem. We have these little electrons, okay, are coming out the end of this electron transport chain. They didn't become a part of this. And so what they're going to do is they're going to become a part of this. They're going to help in the stabilization here, which is in Photosystems 1, which also has reaction center, but this time with chlorophyll A P700. It's still going to have a light harvesting complex around it with the chlorophyll B and the carotenoids or carotenoids. Okay? Everybody's going to get excited also by light energy. It's not like the sun isn't shining on this too. Okay? So, you're going to get light energy, exciting all these guys again. As soon as pigments are excited, they fling electrons. These electrons are going to end up going up here. Uh-oh, I hear my phone. Hold on just a second. Hi, Dad. Yeah, I'm almost done. I just have to finish. I'm almost done with light dependent, and I only have light independent, and I'm out of here. Everything else is done. I've been having video issues, so I'm hoping to be out of here hopefully in about 8 minutes, okay? Okay, love you. I'm so sorry. Love you. Bye. All right, so we've got light energy getting these guys excited. They're losing an electron, but they're being stabilized. Remember over here how the electrons going away meant water got to be split over here at photolysis? Over here, because of the electrons coming in here, no water splitting over here in photosystems 1. So, just like before, electrons, anytime they get flung off, that was not a word. Flinged is not a word. It's getting late. Anyway, so it's getting flung off this lovely chlorophyll A, P700, and goes to another primary electron acceptor molecule. Okay? There, that then goes through again another electron transport chain. They are different proteins than this one. It's a different electron transport chain. But it's the same basic process of going through these and exciting some proteins so that it pops out an electron that can be picked up and put on an electron taxi. NADP H plus H pluses. Okay? So that NADH in cellular respiration, the NADPH in photosynthesis. To be able to get these, you need the electrons from the end of this electron transport chain, as well as some NADP and, of course, some hydrogen ions. So they can then transport some hydrogens and electrons to this next process. So, in photosystems 1, you've got some more excitement going on, still going to a primary electron acceptor molecule, down a different electron transport chain that makes NADPH plus H's to be able to transport those electrons to be able to do the reduction phase of the light-independent reactions, also known as the Calvin cycle. Okay? But that's what we call, so we're going to go like this. I'm going to make a little blue square right here. This is the non-cyclic electron flow of the process. That's if everything's going, you know, A to B to C and everything's going in order and life is beautiful and we're just going through the process of photosynthesis. Sometimes, though, a plant will need to have some extra ATP. If that happens, it can trigger this photosystems to do something different. It can cause it to do cyclic electron flow. Also known in my little brain as self-stimulation, because what happens is the electron that went here to the primary electron acceptor molecule in photosystems 1, this one takes a little different trip. It actually goes back to the electron transport chain that was used in photosystems 2, and it goes transferred from these kind of molecules again. So what that means is it's going to generate the movement of hydrogen ions and the production of ATP instead of going over here and making NADPH. But what that does is that electron goes here, and guess what? It goes around and around, just self-stimulating again and again and again. And as long as it's going in this cyclical flow, you're going to get ATP out of that, not NADPH. Okay? So, let's say it's done the non-cyclic electron flow. We've got ATP and we have some NAD plus H's that are going to go into and help run this process right here, the light-independent reactions, also known as, aka, the Calvin cycle, because that's his last name. All right, so let's talk about this for a minute. There are, in this part, there are 3 phases to this light-independent reaction, okay? There is the first phase, which is here. I'm going to make a little bracket to here. Then there's the second phase, so this is phase number 1. Okay, the first phase is the carbon fixation phase. Okay, so that carbon fixation phase is where some CO2 has to come in, or enters, that would have been more scientific-y, huh? Comes in through the stoma or stomata and that spongy mesophyll. Okay? What happens is this CO2 that comes in from here then has a carbon molecule stripped off of it. When it is stripped off and you add some water and an extremely important enzyme that you have to know the name of, RUBP carboxylase, because it's an enzyme that adds carbon. So notice this carbon here and this 5-carbon molecule, which is RUBP, also known as ribulose biphosphate, because it has 2 phosphates that are energizing it. Don't worry, in IB12, we'll have all sorts of fun with that. Okay? You have this process where it takes ribulose biphosphate, because remember there's a cycle, adds on this carbon using RUBP carboxylase to make this unstable C6 molecule. And it's so unstable during this carbon fixation phase that all you got to do is add some ATP that came from photosystems, right here, from photosystems 2, and some electron taxis bringing in some NADPH plus H pluses. They're going to be bringing in some electrons, okay, to do the next phase, okay? This phase, oh, that's a bad bracket, but you'll understand. The next phase being the reduction phase. And reduction phase, because it is moving those electrons, remember, that came in on the taxi, okay? These ADPs and NADPs, these guys go back and get recycled to the light-dependent reactions. So it's cyclical, right? It takes these and uses them again. We're such good, life is so amazing when it comes to reusing, okay? It's just amazing. So what we have now, we had this unstable 6-carbon molecule that got formed during the first phase, the carbon fixation phase. You take and add the energy from photosystems 2 through ATP and the electrons that were formed during photosystems 1 to do a reduction phase to form G3P, which is glyceraldehyde 3-phosphate. To be able to make this G3P, glyceraldehyde 3-phosphate, it's actually going to take 3 kind of like what we call turns of this visual that you see right here. Because you only have 1, right? You got to do it 3 times to be able to do this. So here is our G3P that ends up out of the process that can then be used to make glucose if it does 6 turns of this, which then can be used to make disaccharides, could be used to make polysaccharides, like, oh, I don't know, cellulose, so a plant can have cell walls, right? That kind of thing, okay? It can also be used, this G3P can be used to make proteins for that plant. It can be used to make lipids that that plant needs, say in its seeds. And it can, of course, be able to be used to make nucleic acids, because, of course, they need to make DNA, RNA, like ribozymes and things like that. So nucleic acids, and, of course, ATP is a modified nucleotide. Remember, an RNA nucleotide. So now we've got what we need coming out, but the problem is we need to cycle this back around. You need to do the last process over here, which is the third phase, which is RUBP carboxylase. No, sorry, excited, want to go home, worried about my dad. Okay, anyway, sorry, try again. The regeneration, you can just skip that part if you want to. RUBP regeneration phase. In other words, you got to make and use some ATP, so some of that ATP from here gets utilized to be able to glue things together to make a ribulose biphosphate, so you can do it all over again. So, when this is all said and done, remember how on this side, we found that aerobic cellular respiration, you take glucose, you get some oxygen in there, and you're going to give off some carbon dioxide, some water, and get a whole bunch of freaking energy? Well, guess what? Photosynthesis. You're going to take some CO2, you're going to take some waters? Wow, that's not a word either. Plus some light energy. And amazingly enough, you ready for this? It's 686 kilocalories of energy to get, okay, C6H12O6 plus, thank goodness for us, some leftover oxygen. Okay? So ready? Watch this. 6 CO2 plus 6 O2s. Oh. Okay? And then look right here. We've got some CO2s, some waters, and some, in this case, energy, 686 kilocalories. Ready? 6 CO2s, 6 H2Os, and oh look, 686 kilocalories from 1 glucose mole of glucose, right? So, by the time we're all done this gorgeous process, we have done, right, in detail, what we did earlier today. If I can find my piece of paper. If I can find it, right? This beautiful cycle within the environment. Okay? So, hopefully that helps. I'm hoping that helps you. I'm going to go home and kiss my children while they sleep and thank my dad. Anyway, have a great night and day, and you're probably watching this during the day. I'm tired. All right, have a great day.